Thank you very much, Fadi, for that introduction and for organizing this webinar. Very happy to be here and participate. And I wish everybody either a good morning, good afternoon, or evening, wherever you're coming in from. So as Fadi outlined, I'm giving the first presentation, one of two, this morning, and for here. And it's going to be on the comprehensive, targeted, and quantitative profiling of the human milk metabolome, specifically using the Quant 500 platform or metabolic kit. So without any hesitations, I'll proceed. Let's see if I can get this going here. There we are. Well, first of all, I want you to just put up a bit of information, which I'm sure everybody is aware of, that the human breast milk metabolome is unique in that it has a number of nutrients and bioactive factors that promote that promote a healthy development and growth for the infant. This is an abundant literature that's been around for many, many years now, decades, and it indicates that there are critical constituents of human milk that can protect against disease and may have a lifetime of benefits. In fact, the human milk metabolome is made up of thousands of molecules, and these are derived from various sources, as shown in this picture here, taken from this very nice publication by Stinson and others. And the milk metabolome can be affected in many ways, from not only what's being produced in the mammary cells, but also from the milk microbiota itself and the gut microbiota, and from the diet, and perhaps also from other tissues which seep out metabolites into the circulation and interact and can cross over into the breast milk. It can be also more complicated in terms of the factors that affect the human milk metabolome. So as we can see here, and this is again from a publication by Fu et al. Recently in 2024, the factors that can affect the milk metabolome are multiple. And what we see here is, they can depend on geographical locations, on the maternal body mass index, pathological state, maternal diet, of course, delivery mode, other perinatal factors, gestational age, and lactation. And these will result in changes in the metabolites that we see over here, depicted over here, which are some of the main constituents, but of course not all of them. And this is just really a summary. And of course, we use mass spectrometry as our main, or NMR as our main technique to identify these metabolites, and bioinformatics to unravel all the information and make sense of everything that we can find. So just to kind of briefly give a little bit of more background here on the methods used. There are both untargeted and targeted metabolomic studies, but the majority of publications are based on untargeted methods that we see, at least in the human milk metabolome area right now, and perhaps in others too. The untargeted platforms really provide the highest number of metabolites in terms of coverage, and but the targeted metabolites tend to focus more on quantitative analysis and on lipidomic analysis, as these are lipids are the major constituents of the human milk. So just to kind of outline a few major reviews here, I'm not going to go into the very deep details that they are, but that are in these publications. So here is a review, a scoping review, database of over 40 publications, and 22 were included in the analysis, and they came up with some main conclusions showing that there are multiple factors that affect the human milk metabolome. Moving on, this study here by Stinson as well, a very nice review on the human milk lipids and small metabolites and maternal and microbial origins. Number of health benefits were identified in these publications. And then by our other guest speaker today, Daniel Hampel and colleagues in 2019, they published one of the first studies there on the biocrates kits, that was the P180, and they were able to identify a large number of metabolites, over 65% of the 188 metabolites in human breast milk. And they made a number of observations there with BMI and so forth. So the next presentation that Will Come by Danielle Hempel is presented, I believe, her most recent study, and I'm not going to steal her thunder on that, so I'm going to leave that up to Danielle to tell you all about that. So, but for this presentation here, what I want to also emphasize is that there are many factors to consider when collecting human milk for metabolomic studies, and they're outlined here, as you can see. These all have various effects. So, the time postpartum milk collected, there are various stages. That can be a big factor. Whether it's manual or pumped, the time of day, and washing of the breast skin, and whether you take the first stream or later during feeding can change. So you can see this can be quite a complex way of trying to understand all the data, especially when comparing across studies. And if everybody is doing it differently, then that really does get a little confusing, and we need to be aware of that and make sure that we're comparing studies that are very similar when looking at our results. So for today's presentation, I wanted to really focus mostly on a study that was published with collaborators at the hospitals in Valencia, Spain, and Barcelona in Spain. And we've collaborated with this group. And this study here was a comprehensive targeted profiling of the human milk metabolome using the Quant 500 biocrates kit. And this was a cross-sectional study looking at the metabolites in terms of delivery mode, breastfeeding practice, and maternal diet. So just to give a little bit more detail here, the study was composed of 123 healthy women. Full fat milk was expressed and stored at -80. And then we used the Quant 500 kit that was supplied by biocrates, and this was done on a Sykes 5500. Now, the time that the milk was taken from mothers up to 1 month of age. Average was 20, and this was pumped, and it was taken every time in the morning up to noon, and the skin was washed, and this was a first feeding collection. And then the milk was stored at -80 degrees until time of analysis. And it was biobagged in Spain and sent to our laboratory here in Dallas, Texas for analysis. So here, just kind of outlining what I've just mentioned, but the kit here also, just to give few more specifics, it measures 630 metabolites and 234 metabolic indicators. We use the MetIDQ program supplied by biocrates. Right now we've transitioned to the WebIDQ, but at the time we were using this MetIDQ program. And then we processed the data with doing batch normalization. This was actually done in about 4 batches. And then we processed the data for limits of detection and filtered the compounds where 50% of the compounds less than the LOD were eliminated from that analysis. And in addition to the metabolite analysis, which is also important here, one of the objectives of the study was to look at the anthropometrical and clinical parameters, and also maternal dietary information, food questionnaires, and intakes of macro and micronutrients using food composition tables. So this was a kind of a very important part of the study here too. So in terms of the metabolites that we detected, we can divide these up into either lipids or non-lipids. Mostly the non-lipids or small molecules are mostly done on the liquid chromatography part, whereas the most of the lipids are done on a flow injection analysis, although we do have some fatty acids here that were measured on the LC part too. So potentially 106 metabolites, small molecules, non-lipids could be detected, but when we found 64 of these, so we were looking at about 65%, 66% of the total here. And then for the lipids, we see these various different lipid classes going down, and the number of potential metabolites that can be measured, which was a total of 524, and we could measure accurately on these 368. So this kind of gives us a total coverage of about between 68 to 69% for metabolites and metabolic indicators in terms of what we can actually detect and pick up. Now, breast milk is a little bit of a challenge in the sense that it has a very wide concentration range for these metabolites, from the low nanomolar up to the high micromolar and even millimolar range for these various different metabolites in all these different classes. And a modification that needs to be done to the kit when using human milk is to add additional calibrators at the low end and the high end in order to capture this complete range in terms of all the metabolites and get good AMR, analytical measurement range coverage. And so moving on from here, we can see in these diagrams the kind of distribution that we find from this study, the samples that were analyzed in this study for both the small molecules and the lipids. And this is pretty consistent with other publications in the literature, where for the small molecules, the largest class of compounds is represented by the amino acids and other amino acid related compounds. And then most of these other classes are making up anywhere in less than 10% in each one, and sometimes as low as 2% of the total. In terms of the lipids, again, this is consistent with the literature, which shows that triglycerides are one of the main classes of lipids in human milk, and that's followed by the phosphatidylcholines and sphingomyelins as the next one, and the glycerolceramides. So we were, looking at this, it was consistent with most of what we see in terms of data in the literature for these distribution of the metabolites. Now, to get to the actual data in the study, what we found is that what was revealed from this study was that there were three main clusters of metabolites, and the data was analyzed in terms of these clusters. There were no real individual amino acids that showed highly significant differences with some of these amperometric or other outcomes in the data that we had from the study. But when we look at we could actually see there were three different clusters that were really quite well defined. And what we see here is that the triglycerols were really quite affected. It was much lower in the cluster 3, and this was by far the one of the most biggest impacts on differences in the clusters. And also for the glycerol phospholipids and glycerolceramides and diacylglycerols and cholesterol esters, they were all we could see these distinct classes. Now, there was also amino acids were also increased. Increased in cluster 3. So when the data was analyzed in terms of these clusters, we could see that mode of delivery was significantly different. So the subjects which had vaginal section were particularly, it was significantly different in the third cluster here, which was significant at the FDR p-value. And when we looked at the associations between the metabolites and the perinatal factors themselves, what we found is here that the DHA levels were significantly different at the p-value. It did not result significant by FDA, but at least for the p-value, we had higher levels of DHA in the breast milk from the mothers that had vaginal births versus the C-section. And in terms of other metabolites for these metabolites for comparing exclusive breastfeeding versus mixed breastfeeding, we see here that the sugars were, represents the sum of sugars that were detected, was significantly higher in the exclusive breastfeeding mothers. And this tends to be consistent with what we see in the literature. There are some reports, similar reports in the literature about that. Another analysis that was performed was to look at the specific dietary components from the food questionnaires, and these are shown that these were associated. These various different classes were associated to the specific milk metabolome cluster. So cluster 1 or 2 or 3 for each of these the different dietary components or dietary classes here. We can see that the blue color here indicates that the metabolome cluster was significantly higher compared to the other 2 clusters. And so in cluster 3, where we had decreased triglycerols, we see that this is associated more with the diet that had more macronutrients and dietary fiber, and again for vitamins and minerals. And there is some increase in cluster 1 here for the total carbohydrates and digestible carbohydrates here, as well as for B12 in cluster 1 in the minerals and vitamins. So there was distinct associations with these metabolome clusters and these various different food categories here. In terms of the associations between the human milk metabolites and maternal dietary patterns, again, we saw some interesting associations, particularly when we do the groups. The subjects were divided into those that had a higher vegetal protein versus a high animal protein, and this again could be kind of expected as we see here that amino acids, sphingolipids, and glycerolceramides were much higher in the subjects that had the high animal protein and high lipid intake. So we could see that this translated over into what was expected as what we would think would happen. But it's nice to have that confirmed. So to summarize these observations from the study, we were able to show that within the sample population, the statistical analysis revealed these different clusters, distinct clusters, and triacylglycerides were the main metabolites showing the greatest difference. And it suggests that this type of lipid, which represents the majority of the lipids in human milk, is highly variable. We didn't see any significant impact on mode of birth on the global metabolite profile, although we found that the C-section mothers had a higher, was associated with a lower DHL concentrations in human milks samples compared to women who had the vaginal delivery. And this also agrees with what we know in other reports in the literature. And hexoses were also higher in the women giving exclusive breastfeeding compared to those on mixed feeding with formula. And this also has been shown in previous reports that human milk lactose is positively correlated to the volume of intake in infants on exclusive breastfeeding regime, and which is actually higher than when you're feeding with formula milk. So it's also well established that the diet affects the composition of the human milk. That's not a surprise, and certainly not for us here in this study. And we observed that the 3 metabolite groups, the glycerol, ceramides, sphingolipids, and amino acids, tended to correlate with a dietary pattern that had a higher animal protein and lipid intake compared to those that had a vegetal protein and fiber. So this analysis was done, as I emphasized in the previous slides, with the MxP Quant 500 kit from biocrates. And these are all the metabolite classes that were identified. And the numbers in parentheses indicate the numbers of metabolites for each class that were detected. At the time, this platform was available for the analysis. However, since then, and it's been available, I believe, for just over 2 years now, there is an expansion plate for this platform, and the kit for this kit is called the MxP Quant 500 XL, and it expands the coverage to 1,019 metabolites and 474 metabolic indicators, which are made up of sums and ratios of metabolites. And this is a kind of quite a significant increase that really is specific to these lipid classes. So it adds these numbers of metabolites in these various lipid classes. And I think this would be very nice to go forward with in future studies, especially in the human milk and in others. it does provide a greater coverage on the lipid profiles. We ran a pilot study with 7 milk, and actually we've done more studies since then, but I'm showing you here just the analysis using the Quant 500 XL on 7 human milk samples. And this just shows the distribution of the proportion of the metabolic classes of the detected metabolites, we can with this kit. And again, this changes the proportions a little bit to what we've seen before in an earlier figure with the Quant 500. Really important ones here are the phosphatidylethanol-amines and the phosphatidylcholines, which the phosphatidylethanol-amines were not detected before, and we can pick them up now in the Excel kit. And there are a number of other important lipid classes and lipid species that can be detected now using this kit. And I think this would be good to go forward in future studies comparing human milk to formula milk. And we have studies already going underway in collaboration with Dr. Sharon Donovan at the University of Illinois. And would be good to keep, looking into these longitudinal studies and examining the composition of the milk at different stages of lactation, and also looking at multiple factors, including other perinatal factors, diet, caloric intake, medications, and also with the microbiome. And importantly, making these associations with infant growth and development in terms of the composition of the milk metabolome. I'm going to take just one more slide to show that there is also another interesting publication that we published in 2023, because there are 2 metabolites that are not detected in the Quant 500 Excel or Quant 500 kits, and these include S-adenosyl-methionine and S-adenosyl-homocysteine, and the reason why I'm pointing this out is that these are important metabolites, specifically S-adenosyl-methionine, which is a methyl donor to over 100 or more methylation reactions, and it donates a methyl group to various DNA proteins and amino acids and lipids. And we use a targeted customized LCMS assay for these panel of metabolites, and what we found is that this metabolite, which is very important in terms of methylation, is over 40 times higher than in maternal plasma or any cord plasma or other biological fluids, and it's absent totally from human milk formula. So this is a kind of a deficiency perhaps here, but these high levels of S-adenosyl-methionine in human milk may be required for the high rate of lipid biosynthesis, particularly from phosphatidylamine to phosphatidylcholine, and could influence other lipids as well that are in the milk. So with that, I think I've probably reached my time limit, and I thank you for your attention, and I'll be happy to take any questions at this point. Thank you.